Hello guys and welcome back to Excel Chat Academy. So till now in the active solute transport, we have discussed the various types of transporters, right? Now we are going to discuss something about the pore forming toxins. So these toxins are the ones which is actually responsible. For example, हमने cholera toxin सुना होगा. We have heard about the anthracis toxins, right? So how these bacteria are actually affecting us. And again, it is all totally dependent on these transporters only. At the end, kya hai? They are the transporters which is helping in the, so for bacteria, it is their survival mode. Then how are we going to overcome this problem? This is what we are going to discuss in this session. Okay. Now, if you see this example, the figure which is shown just over here, you can see that beautifully like a flower like arrangement it is given, correct? And it makes a proper pore over here at the center. As you can see in this case, see all these proteins are there, right? But eventually they come together in the transmembrane region. They are making like a channel through which it is easy for the molecules or the cytoplasmic molecules to be leaked out. Correct. Now here different types are there and it is all important for you guys to understand and remember. Okay. For example, bacteria like Clostridium and Staphylococcus produce proteotoxins that make numerous unregulated pores in the membrane of the target cells to kill them. So यानी कि क्या? These are the bacteria which release this toxin. And again, what are these toxins? They are nothing but proteins only. What is the chemistry behind? They are the proteins only. Now, there are different types of pore forming toxins. Okay, PFT means pore forming toxins. For example, alpha pore forming toxins example is cytolysine. Okay. Beta pore forming toxins hemolysin is there. Another example is Panton, Valentine, Leukocidin. इसमें से आप ये याद रख लो because ये हमेशा पूछा है. Then binary toxins, anthrax toxin, edema toxin, cholesterol dependent cytolysine. So this is again when you say cholesterol dependent, you can talk about here the lipid rafts, if you remember, right? So there the concentration of cholesterol is more. So then these toxins, the cytolysins are totally dependent on the cholesterol. So example is pneumolysine. Small pore forming toxins, their example is gramicidin A. So in this session, all these different types of the pore forming toxins we are going to discuss. Okay. This is something you have to remember. Now, each example we will be discussing in detail. Okay. Now, alpha pore forming toxin example is cytolysine. तो organism क्या था इसमें आपका E. coli. So यहाँ पे आपसे क्वेश्चन पूछ सकते हैं like we have asked in the interviews also कि E. coli तो बहुत ही general bacteria हो गया, correct? It is present in as part of your gut bacteria also. Okay. Then how come it is not affecting or how come it is not affecting the human cells? इसका क्या आंसर होगा? आप जो पानी पीते हो कभी कभी जो बाहर का टैप वाटर पीते हो, in that also E. coli is present. Yes. इसका क्या आंसर होगा?
Maybe the gut is such a place where it can't affect because of all of the other, there's a lot of proteolytic activity already in the gut, right? So because of that.
No, थोड़ा सा और सोचो, but I think you're going correct. थोड़ा सा और सोचो.
Maybe the gut is affected, but then the cells replace a lot faster, right? In the gut.
Okay. Not exactly. See, then if these bacteria, okay, which are producing alpha pore forming toxins and all, the thing is, first of all, we have this brushing away activity of our cilia, which is present in the intestine. ठीक है, एक तो वो रहता है आपके पास that you remove E. coli, you don't allow the bacteria to get settled in the gut. ठीक है, and even if you have this thing, we, as you said, we have become resistant to that. Plus, the E. coli which releases these toxins, okay, they are like what to say, a group of those bacteria which of course you are not going to drink the bad water. कितना भी गंदा दिखेगा, even if you're so thirsty, you're not going to drink unless until you see that it is plain water, it looks clean, बिस्लेरी बॉटल में है, but you don't know that उसमें E. coli है या नहीं है, ठीक है. So we have got different strains of E. coli. So each strain has got difference in their toxin production, and हर E. coli आपका toxin produce भी नहीं करता है. So the water which we take or what is present in our gut, those are not the ones which is going to produce the toxin. समझा क्या? मैंने थोड़ा complicate कर दिया. Let me say it again. It's just the E. coli. E. coli में ये आपका species हो गया. E. coli में you have got thousands of strains. Okay? ये समझ रहा है species and strain का अंतर? Are you guys familiar with that at least?
Yes, ma'am.
हाँ? इतना अंतर आपको पता चल रहा है, right?
Yes, ma'am.
Okay. Okay. So for example, मैं आपको एक example दे सकती हूँ that if I have pathogenic and non-pathogenic, okay, let it make it more clear for everyone. So these are the two categories of organisms, ठीक है. Pathotype बोलते हैं हम इसको. आपने micro में, I don't know whether it has been taken or not. So we say it as pathotype. Pathotype means whether, for example, E. coli is pathogenic or non-pathogenic. ठीक है. Now under pathogenic we have got many strains. For example, we say enteropathogenic E. coli strain is 126 nm. ये आपका strain number हो गया. Species क्या हुआ आपका? E. coli. ठीक है. Strain हो गया आपका 0126 जो भी number हो गया. Similarly, we have got another species like 157 हो गया या 1493 हो गए. So these are the different strain number of E. coli which are pathogenic. Okay. Now coming to the non-pathogenic. So non-pathogenic E. coli is also present. For example, strain name is K12. So अगर आपके पास ये present है, so it's okay. These are present in your gut. So they are not going to produce the toxins. Okay. Pathogenic E. coli are going to produce the toxins. अब समझ में आया? So cytolysine जब मैंने बोला है, it is a species E. coli, but which strain it is going to produce वो mention नहीं किया है. So what we need to understand is that E. coli species में there are certain strains which are pathogenic. Those pathogenicity is coming from the ability to produce the alpha pore forming toxins. अब समझा?
Yes, ma'am.
Huh? Clear to everyone?
Yes, ma'am.
Okay, cool. Okay, so this is how the categories are given. ठीक है, ऐसा नहीं कि हर E. coli खराब है. You just have got the pathogenic and the non-pathogenic. Okay, so then similar for the other cases also, pneumonia and ये सब. ठीक है. Okay, let's proceed now. Now we have got the alpha pore forming toxin cytolysine is there. Okay. Now in this case, okay, in the case of this cytolysine, let's start. Okay. So first you have got the inactive subunits. LLLS. Okay. Now before we proceed over here, ये आपको कहाँ से आ रहा है? ठीक है? Here the genes are getting expressed. So these are the genes which is getting expressed. For example, MBAI. Okay. Now when these genes are getting expressed, what is happening? ये जो मेम्ब्रेन बनाया है ये आपका बैक्टीरिया है, ई कोलाई का बैक्टीरिया है, ठीक है, सॉरी ई कोलाई का मेम्ब्रेन है. नाउ ई कोलाई अंडरस्टैंड्स दैट देयर इज अ ह्यूमन सेल, ओके, ह्यूमन टारगेट सेल इज देयर, आई नीड टू प्रोड्यूस साइटोलाइसिन. Okay, now cytolysine is composed of two subunits, L and small, that is large and small. ठीक है. Now, when R is for the regulation, okay, when it is required or not required. So what is happening? So your large subunit and small subunit gets activated. So those are produced, okay. Now, M is required. It is those enzymes which is required for the post-translation modification on these subunits, correct? For example, it can be, if you see in the structure, it can be change in the secondary structure, or it can be making like from what to say, the from inactive form to active form by degrading the starting of the peptide, which are like signals only, correct? अगर जैसे ये एक polypeptide chain है, starting में एक signal रहता है, so there are certain enzymes which actually remove this signal and allow them to get modified into their respective secondary or tertiary structure. Okay, this is that is what is meant over here. So you have got the cytolysine precursor subunits, large and small. Then this M, it is releasing certain enzymes which is going to do post-translation modification on them, resulting into the formation of modified subunits. Now these modified subunits, they have got the signal which through B, okay, through B. So B यहाँ पे क्या हो गया आपका transporter, okay, B आपका transporter हो गया, which is coded by this particular gene. So it will help in the transport of the subunits large and small outside. Obviously, right? E. coli कैसे act करेगा जब वो toxin बाहर produce करेगा. Now they go outside. So your inactive subunits, large and smalls, are now secreted out. Correct? Now you have got the another enzyme. So these is proteases. Proteases क्या करेंगे? They are gonna chop off these inactive subunits, converting them from inactive into active subunits. So you can see this is doing the activation. So again, there's a certain changes in the structure and you get the active toxin subunits. So once they become active, the small and the large subunits come together and then they get integrated into the membrane of your human or the target cell. अब जब ये integrate हो जाएगा तो what will happen? All the cytoplasmic content of the human cell will come out. ठीक है? This is how alpha pore forming cytolysine is functioning. So this is the mechanism. Okay. Any doubts, guys, or do you want me to repeat again?
Ma'am, which channel does the protease use to come out of the E. coli cell?
This one, the protease you are asking, this
Yes,
one?
yes.
See, again, for each particular component of your cytoplasmic proteins, if it is supposed to be secreted out, it gets a signal. ठीक है? एक second, huh? So this again, once we complete the transport, membrane transport, then next topic is this only, like how one particular protein is targeted for the extracellular release और it is meant for the intercellular integration. जैसे कि अगर ये membrane में bound होना चाहिए या माइटोकॉन्ड्रिया में बाउंड होना चाहिए, वो कैसे एक सेल डिसाइड करता है, ठीक है? नाउ जस्ट फॉर एग्जांपल ओवर हियर, इफ यू टॉक अबाउट दिस पर्टिकुलर प्रोटीएज, सो इट विल हैव अ सिग्नल for the release outside of the membrane. ठीक है? So let us say that this is a protease and here the signal is present. Okay, so there are certain transporters like B which actually remove the signal. So you have got the signal peptidase. Okay, so this signal will be removed. This peptidase, this peptide, okay, the signal will be removed. This particular enzyme will be then transported out, and for them there are two ways mechanisms are there. Either they will have the transporter, or if it is in bulk, then they will be exocytosed. So the process of exocytosis. Clear, Shruti?
Yes, ma'am. Yeah.
Okay. Okay. Anything else do you want to ask? Anyone else? This entire mechanism. तो आपको शुरू कहाँ से करना है? यहाँ से शुरू करना है. You need to understand then when there is a regulation. Okay. Regulation मतलब क्या हुआ आपका? अगर आपके पास target cell नहीं है, then there is no need of required of, or there is no requirement of having these large subunit and small subunit cytolysine to be produced. तो यहाँ पे वो बंद हो जाएगा. Now when you see दुश्मन सामने है, human target cell is there. So it says that I need the expression of these particular genes, correct? M चाहिए मुझे मॉडिफिकेशन के लिए, B चाहिए मुझे ट्रांसपोर्टर के लिए, A मुझे चाहिए प्रोटीज एक्टिविटी के लिए, I क्या है आपका इंड्यूसर होगा, सॉरी इम्यूनिटी, वाई इम्यूनिटी ये आके मेरे ही मेम्ब्रेन में आके अटैक ना करे, नहीं तो फिर मेरा कंटेंट चला जाएगा इफ आई एम द इक्वल आई. सो दिस इज प्रोटेक्टिंग itself from its own attack. समझा? This is like a shield.
Yeah, one more thing, ma'am. How does it recognize that there are human cells nearby?
So there comes again your cell signaling. So it understands, if you remember, we have talked about in the RBC, there are certain lipid rafts are there. These lipid rafts have certain integral proteins which is acting as a receptors, right? So these receptors means what? ऐसे कुछ sugar molecules रहते हैं, correct? So these sugar molecules are interacted with the bacteria. जब वो बैक्टीरिया को समझता है, then they understand that ये particular human cell है या eukaryotic cell है, let us attack them. Because these won't be present on the next E. coli जो उसके बाजू में होगा. So it understands अरे ये अपना ही भाई है. So they don't attack on the E. coli next to it. समझा? Like for example, in humans we see, right? We see directly and understand ये human है या ये animal है, right? In the case of E. coli, it is the receptor ligand interactions which gives them the signal. Okay, ये हमारा receptor नहीं है, ये human का है. This is my target. I should attack. Otherwise, they are gonna attack me. See, अगर वो macrophage होता या कुछ और होता, it is going to engulf the bacteria. उससे पहले bacteria को counter attack में क्या करना होगा? They should start releasing these cytolysine to protect themselves. Correct?
Okay, ma'am. Yeah.
I mean, I know it is very complex, but धीरे धीरे सब समझ में आ जाएगा. Once you understand everything, then you just have to connect the dots. Okay. So whatever we have discussed is the same thing written over here. Okay. We'll just go through it. So it is a model for the expression of the cytolysine from the E. fecalis. The toxin subunits large and small are ribosomally synthesized in the precursor form. Then are post-translation modified by the M, which introduces modification characteristics of the antibiotic class of bacteriocins. Okay. The modified peptides are then further processed and secreted by B, which serves as both ATP binding cassette transporter and a signal peptidase. Okay, so here you got the point. See, abhi we have recently talked about how ABC are working. So here your B is acting as a ABC transporter plus signal peptidase. इसका क्या मतलब हुआ? जैसे ही यहाँ पे B है, okay? Your ABC transporter is there. The moment those small and large subunits were coming, whatever the signal they had, because ये आपको बाहर pump out करना है, बाहर extracellular release करना है, right? So those signal will be removed. So that also will be done by the B. Okay. The matured externalized peptide subunits L and S undergo activation by A, the proteolytic cleavage, which removes the identical 6 amino acid residues from the N terminus of each subunit and generates the active toxin. Now, I confers self-immunity from the bactericidal action of the cytolysine. Then the expression is regulated by R1 and R2. Okay, the same thing what we have just discussed now is written here. Okay. Now coming to the beta pore forming toxins. So अभी जो हमने डिस्कस किया था वो आपका अल्फा था, ठीक है? Now why alpha? Because if you see the structure वो अल्फा हेलिकल रहेगा, ठीक है? यहाँ पे आपके बीटा पोर्स आ रहे हैं, बीटा स्ट्रैंड्स आ रहे हैं. So beta pore forming toxins are dimorphic proteins mostly made up of beta strands based domains that are soluble monomers and assemble to form the pore. अभी क्या बोल रहे हैं ये लोग? That it is a soluble and it is a monomer. तो यानी कि once it is required, then only they assemble to form the pore. Okay. These pores disrupts the regulated gradient of ions and small molecules of cytoplasm, allowing continuous outflow along with the nucleotides and amino acids. So what they are saying is that एक बार आपका यहाँ पे ये पोर बन गया तो साइटोप्लाज्मिक आयंस विल गो आउट, साइटोप्लाज्मिक न्यूक्लियोटाइड्स एमिनो एसिड्स विल बी थ्रोन आउट, जिसकी वजह से क्या होगा, द सेल विल बी डिहाइड्रेटेड, द सेल विल नॉट हैव इट्स न्यूट्रिशन एंड इट इज गोना डाई. It also facilitates excess diffusion of water into the cell that leads blebbing or swelling, which results in the cell death. So either way round. Okay. So here it is. Wow, ऐसे देखते ही लगता होगा ना कि क्या है ये चीज. But those all are very interesting, guys. Okay. Can you see PFT, pore forming toxin? So they are first the insoluble ones, right? So they are the so not insoluble, they are the inactive soluble PFT, right? छोटे छोटे से हैं. Now they get the receptor or the lipid, जो यहाँ पे भी आपका lipid raft होगा, okay? They come and they bind to that. Now once they bind, what is happening? So these inactive PFTs are secreted by your organisms. ठीक है? They come and interact with the lipid rafts containing receptors of your host cell. ये आपका host का है membrane. उसके protein receptor पे आपका ये inactive soluble PFT bind होता है. Single. Later what happens is that they come together, okay, and then they get inserted into the membrane. Can you see? They are getting inserted into the membrane. So what is happening? The pore is present, eventually resulting in the loss of ions, loss of amino acids or energy. Is it clear, this diagram? Should I repeat again? Tanvi, is it clear to you?
Yes, ma'am.
Okay, cool. Who can explain me then this? Tanvi, would you like to try?
Okay, ma'am. So at first, this inactive soluble PFT comes and bind to the lipid receptor over the host membrane,
Correct.
and then it insert in the membrane and make a pore structure, and therefore the loss of these ions takes place in the host
Yes,
membrane.
absolutely correct. Okay. So I hope everyone got Shruti and Akshaya. It's clear to you guys also, right?
Ma'am, just one more thing. The pore that is formed, that will only release the ions and proteins until the equilibrium is formed, right?
No, see, ये cell का नहीं है, ये outsider का है. So it doesn't have the what to say? It doesn't have the responsibility कि equilibrium ही बनना चाहिए.
Nee, but as soon as there's more solute outside, it will stop, right? Because
no dear, see, I agree. अगर बाहर अगर आपका वो present है तो obviously plasma membrane is gonna act for the osmosis, तो आपका वो equilibrium reach होना चाहिए. I agree. But when an organism is attacking on you, वो क्यों attack कर रहा है? Equilibrium achieve करने के लिए नहीं करता है ना. It is going to use all your ions, all your amino acids, all the proteins, what is being released from the cytoplasm.
Okay.
है ना?
So what happens is that the organism outside will use up your protein so that it never reaches equilibrium.
Correct. Okay. अब देखो, अब मैं एक और चीज मैं बताती हूँ. Good, you asked this question. This is your host cell and these are the bacteria. ठीक है? So when they attack, it's not that only एक E. coli एक pore release कर रहा है. So one E. coli will be releasing so many molecules of the toxin. Another E. coli is there, it is also releasing so many molecules of toxin. जब एक साथ इतने सारे toxin attack करते हैं, then the host cell is they start losing the cytoplasmic content and all these bacteria which are present, okay, because of the chemotactic activity and all, they will come here, they will start eating all those things, है ना? Because they are going to divide eventually after 20-40 minutes. समझा? तो यहाँ पे कभी भी इक्विलिब्रियम वाली बात नहीं आएगी. It will be used up and the cell will die. Clear?
Yes, ma'am. Yeah.
Huh? Good. Okay, anything else? Okay then. Okay, so now let's move on to the next one. So binary toxins. So we have covered alpha pore forming toxins and beta pore forming toxins. Now coming to the binary toxins. So it consists of component A and B, where A is the enzymatic component and B is the membrane altering component. Okay. Component B, it forms homo oligomeric pore that helps in the entry of the component A in the cytosol where it is going to inhibit the normal cell process. ठीक है, एग्जाम्पल हो गया आपका एंथ्रैक्स टॉक्सिन. तो अभी क्या कर रहे हैं यहाँ पे, दो कंपोनेंट हो गए. ए क्या करते हैं, बी आपको हेल्प करेगा मेम्ब्रेन के अंदर जाने में. किसको, ए को, सो दैट दिस एंजाइमेटिक कंपोनेंट ऑफ द टॉक्सिन इज गोना स्टॉप द नॉर्मल सेल प्रोसेस. ठीक है. नाउ this anthrax toxin, this is really very interesting and important. Okay, so anthrax toxin, so it has got the it is also known as the exotoxin. It is secreted by virulent strains of the bacterium called Bacillus anthracis. इसके बारे में कहानी आपने सुना ही होगा, right? This anthrax जब फैला था चाइना में. Okay, so it is composed of three components. The cell binding protein, which is known as the PA, protective antigen. Guys, थोड़ा सा याद रखो because in the figure it will be helpful then. PA is a protective antigen. This is going to help in binding of the cell. ठीक है. Then there are two enzyme components called edema factor and the lethal factor, EF and LF. Okay, so two components होते हैं आपके, एक membrane binding और एक enzymatic. तो यहाँ पे आपका PA कौन से कंपोनेंट में आ रहा है, इज इट A और B?
A.
No, B, because B is your membrane binding component. ठीक है. So protective antigen is cell binding protein where A is the enzymatic. So A में आपका आ रहा है edema factor as well as lethal factor. Okay, now all these three together, they are going to come together to have the toxin activity. Okay, same thing. So they have shown, see, protective antigen, if it is there, it is going to help in binding of the membrane, correct? Then only the lethal factor will come. Now suppose lethal factor is coming, it's a lethal toxin, it is going to make sure the it is going to make sure that the target cell is dead. ठीक है. Now with the protective antigen, with the edema factor, again it is going to make sure it is the cell, the target cell is going to swell. ये edema toxin है, right? It is edema resulting in the edema of the cell. Okay, now अभी आपको इसमें क्या याद रखना है, that if you don't have the PA, protective antigen, then the lethal factor or the edema factor cannot do anything. तो इन दोनों के लिए आपको PA की जरूरत है. Lethal factor is the enzyme endopeptidase, whereas edema factor is your activating adenylate cyclase. Okay, now let's see the mechanism of all the three factor coming together. So they assemble the complex containing the toxin components are endocytos. इसका मतलब क्या, ये तीनों को host cell जो है endocytos से अंदर ले लेता है. ठीक है. Now this endosome, so whatever its form, let me just draw for you guys. Okay, this is how sorry, यहाँ पे आपका bind हुआ है these factors, so it induces the host cell to form the endosome like this. So क्या होगा, ये component आपका ये cell के अंदर आ गया. ठीक है. So this becomes your endosome. Okay, this is how the toxins are translocated inside of the target cell cytoplasm. Now once it is in the cytoplasm, the enzymatic components of the toxin will disrupt the immune cell function. यानी कि it is going to disrupt the cellular signaling and the migration. So it will make sure that the target cell is not able to give any response to the neighboring cell. ठीक है, help के लिए वो नहीं बुला सकता फिर. Anthrax toxin allows the bacteria to evade the immune system, proliferate and ultimately kill the host animal. तो ये तो हमें पता ही है, so it's very deadly. Okay, now this is the mechanism. So your Bacillus anthracis, it's like it's a capsule. It forms a capsule whenever there is a unfavorable conditions. So they release these exotoxins. So exotoxins consist of your protective antigen, lethal factor, and edema factor. Okay, so they all these factors, they go and bind to the receptor of your host cell. Okay, and they have mentioned it is the furin. Okay, so they come and bind together. You can see that with the help of the PA, they come together. Okay, your PA binds to the receptor. ये receptor जो है वो आपका host का है. To this light gray cream color आपका PA है, this one. Okay, they come together, right? And it makes sure that the lethal factor and the edema factors are binding and getting endocytosed. So once it is coming inside, okay, once it is coming inside, edema factor is going to activate or hyperactivate adenylate cyclase, which will convert ATP to CMP, resulting in the edema of the cell. Now, if it is a lethal factor, it is going to affect the cell signaling pathway of your cell, ultimately resulting into the death of the cell. Is this clear, guys? The mechanism is clear.
Yes, ma'am.
Huh? To everyone? Almost, thoda bahut. Okay. अभी कैसे होता है कि how the bacteria and also if you remember there was this case that on the paper, ठीक है, ऐसे पेपर में powdered form of Bacillus anthracis was taken. तो ये कैसे कैप्सूल ले लेते हैं, कैप्सूल फॉर्म ले लेते हैं these bacteria whenever the conditions are not favorable. So it was powdered, okay. So in the paper it was taken and राह चलते लोगों के ऊपर ऐसे blow कर दिया था. So what happens? All of a sudden you are gonna breathe in, correct? So these bacteria in the powdered form was inhaled by the people, and these capsules started releasing the exotoxins, and the people started, coughing and dying because of this. Okay, so this is how it became the Bacillus anthrax, or the anthrax toxin resulted in the formation of आपने सुना ही होगा bioweapon करके. Okay. So this is how, this is also a terror. अगर आप गोली से नहीं मार सकते तो आपको फिर ऐसे. There was this case also जहाँ पे there was some bug. Okay, मुझे भी वो bug का नाम याद नहीं आ रहा है. So it was inside the what to say, inside the person. Okay, and एक तरह से वो अल्ट्रासाउंड जनरेट करता था जिसकी वजह से humans were always thinking that something is as a buzzing around, okay, and that was making them irritated, but actually those bugs were getting replicated inside the body, okay, multiplying inside the body, and when the person died, from all the pores of the particular person, these bugs came out flying and infected the other people around. So ये भी एक तरह का terrorist attack हो जाता है, ठीक है. So that time this bio weapon people were very interested, like this is better, and अगर कल को आप पकड़े भी जाओगे, like unless until you get caught and all, it is difficult to what to say, ऐसे case डालना कि आप एक bug के वजह से मरे या किसी bacterial attack की वजह से मरे. Since government now understands the importance of the science contribution in this, it has been considered as a bio weapon now. ठीक है, अगर राह चलते अगर कोई बंदा बैक्टीरियल इन्फेक्शन की वजह से मरता है, यू कांट फाइल अ केस फॉर दैट, करेक्ट. अभी सिर्फ एक के साथ अगर ऐसे हो, बट इफ देर इज अ मास पॉपुलेशन, जैसे कि पूरा का पूरा इंडिया गायब या कोई अदर कंट्री गायब हो गया जस्ट बिकॉज ऑफ दिस इन्फेक्शन, देन इट कम्स अंडर द कैटेगरी ऑफ बायो वेपन और ऐसे बायो टेररिस्ट. Okay, this is not required for the exam and just giving the extra information. Now, the fourth category is the cholesterol dependent cytolysins. So there were 5 if you remember, right? So now cholesterol dependent cytolysins, they are the family of beta barrel pore forming exotoxins secreted by gram positive bacteria. अब तक जो हमने डिस्कस किया था वो gram negative था. Bacillus is your gram positive, but okay. E. coli is gram negative. So they are secreted as water soluble monomers that when bound to the target cell form a circular homo-oligomeric complex containing as many as 40 monomers. Examples are nemolysins. Now, okay, so sorry. Now look at the structure here. If you have got 30% of cholesterol, can you see it? So in the lipid raft, you are going to have the maximum concentration of the cholesterol. अब ये cholesterol dependent toxin है. So what is happening? So these pneumolysine or this particular toxin will make interactions with the cholesterol. See, can you see it over here? So they are making interactions with the cholesterol so nicely that they get integrated into the host cell. If you had 0% cholesterol, then it is difficult for this particular pneumolysine to interact and cause any damage to the particular host cell. Okay. So that's why it is called cholesterol dependent toxin. So it can lyse and interfere with the function of the cells and soluble molecules of the immune system. It will aid in the bacterial during colonization by facilitating the adherence to the host, invasion by damaging the host cells. But overall, what is happening is that what you need to remember is that you need pneumolysine to be active only if you have got the cholesterol. Okay, is this part is clear? The dependence on the cholesterol. This figure is very clear in order to understand. Yeah, everyone, Akshaya. Okay. Now again, same thing. Okay, so you have got the lipid raft because there only you have got the maximum of your cholesterol, right? Now what is happening is that these lipid rafts are present and these molecules of your pneumolysine, PLY जो दे रखा है, pneumolysine, whenever these lipid rafts containing cholesterols are there, they come together, they form a complex. Okay, and eventually results in the formation of the pore over here. Now, how the host is gonna repair itself? Okay, एक वो भी important है कि अगर आपने पोर तो बना लिया, ऑर्गेनिज्म ने टॉक्सिन पोर तो बना लिया, हाउ एज ए होस्ट आई एम गोना एस्केप फ्रॉम दिस अटैक. समझा? अब क्या करेगा होस्ट? सी, you see that the slowly these pore forming units are coming to the plasma membrane, they form the pore and it eventually results in the loss of the ions from the cytoplasm. Correct? Now how I'm going to repair it? As a host, in order to repair, what I can do is जहां जहां पे ये present है, these molecules which results in the formation of the pore, what I can do is मैं उतना pinch off करके अपने membrane से ही हटा दूंगी. समझा? So I'm gonna remove this much of vesicle. So instead of dying, only this much part of plasma membrane is being lost, which I can recover with the proper nutrition available. So instead of killing or instead of getting murdered by these bacterial pore toxins, it's better that I pinch off this much part of me and get rid of these pores. Beautiful, right? Yes. Samjha? Shruti, Tanvi?
Yes, ma'am.
Yeah, this is really very simple, right? Now, as I mentioned, you have got the cell, ये bacterial है and attach यहाँ पे रहेंगे. So, see, the size of the protein and the size of the plasma membrane you have to understand. अभी मेरे पास ये अगर है तो I'll just pinch off and remove this. Pinch off and remove this. Pinch off and remove this. Okay? Of course, the size is gonna get small, but it is going to be alive. Okay? So this is how host is protecting itself. Now the last one, small pore forming complex. So example is gramicidin or gramicidin D. It is a mix of ionophoric antibiotics. Okay, example is like gramicidin A, B, C. So which is made up of 80%, 5%, and 15%. So ये क्या है? Okay, these are extracted from the Brevibacillus brevis. They are the linear peptides with 15 amino acids. So कैसे होता है कि बहुत सारे छोटे छोटे पेप्टाइड आपके रिलीज किए गए, ठीक है, by the organism. So these small peptides will come together to form the pore. I'll show the structure. Okay, so this is the structure. Can you see? ये सारा आपका peptide structure है. Okay, this is your amino acid structures only. What do we see over here is that they are released as a linear peptide, and so many it will be released. So these small peptides come together, they get integrated into the membrane like this. Okay, they come and they get arranged like this, eventually resulting in the formation of the pore, जो आपका ऐसे लॉस में या वाटर इनटेक में रिजल्ट कर सकता है. दिस इज वन एग्जांपल. ओके, क्लियर? The mechanism. So they behave like an ionophoric substance. They self-associate and thus form small pores which causes the leakage of essential cations from the cytoplasm. So if it is monomeric, it cannot function. देखो, जब यहाँ पे present है, it cannot allow the passage from here. If it is present here, it cannot allow the passage from here. But when they self-associate like this, okay, they result in the formation of the pore, which will result in the loss of the ions from the cytoplasm. Yes. Any doubts over here, guys? That's all. So these were the 5 different types of toxins in which we have discussed about the alpha, beta, binary toxin, in which the example was anthrax. Then we have talked about CDC, that is cholesterol dependent cytolysis. And the last one was your small peptide forming the toxins, for example, gramicidin. Okay? Is it clear, guys? The toxins which is formed. Similarly, the venom which is produced by the snake, they are also peptides, small peptides. Okay, वो ऐसे ही peptides होते हैं जो self assemble होके results in the loss of the activity, and they are so strong that they go and bind to the pain nerve endings, because of which we feel a very intense pain in our body. Okay, yeah. Anything else? Any doubts, guys? You want to ask? Akshay, Tanvi, Shruti?
Ma'am? This gramicidin, when it self-associate, will it be lethal to the cell?
Yes, see, if you start losing all the important cations from the cytoplasm, eventually, if it is not being rectified or repaired by the host, then it is going to be lethal to the organism. Correct?
So it is a matter of chance that whether it exists in monomeric form or the dimer.
No, see, not chance event. When an organism is there, it is releasing. As I said before, also, वो एक या दो release नहीं करते हैं. They release in so many multiple copies. It is just the timing that how fast they self-associate and attack the eukaryotic cell or any other target cell. अगर उसके टारगेट होने से या उसके पहले सेल्फ एसोसिएट होने से पहले, if the eukaryotic or the host cell understands and kick off the that part of the plasma membrane, then it is going to save itself. It's just how fast the host defend itself or the attacker is fast. ऐसा है, कौन सबसे पहले क्या अटैक कर ले या डिफेंड कर ले.
And is it prescribed by any doctors, like for any action of any bacteria or kind of?
What, the gramicidin?
Yes. Because it is an drug.
Yes, of course it is prescribed by the doctors, because see, this bacteria is, suppose let us say that अभी हमने बोला Brevibacillus brevis produces gramicidin, so I can use the same thing on the different bacteria to kill. right? It's not always a host cell. I can use this antibiotic to kill the other bacteria.
But won't that be indiscriminate? It will also end up killing human cells.
What?
It won't be targeted, right? It will be killing human cells as well as the
No,
bacteria,
so
right?
that was just the example. Now, if I talk about gramicidin, okay? If I talk about gramicidin, it is specific, okay? Guys, this is specific against the gram-positive bacteria. We are not gram-positive. तो वो हमें नहीं मारेगा.
Okay.
Acha, so I mean, what I meant by the toxin is that these are the 5 different categories of toxin, of which few are lethal to us. For example, Bacillus anthrax ka toxin is lethal to us. Now, when I talk about gramicidin, it is toxic to the gram-positive bacteria. Clear?
Yes.
Yes, ma'am.
Okay. Anything else? So I can then stop here, and in the next we can discuss the. I'll start with the next topic then. Okay.